So hello everyone, welcome back to again my channel. So today in this video we talk about the new video or new topic, that is Bombay phenotype. Okay, so I write here the Bombay phenotype. This is also called Bombay phenotype, or you like to say it's also called Bombay blood group. Now we have to understand what is Bombay phenotype and Bombay blood group. Basically, we all know that we have 4 types of blood group, right? We all know, I write here A, B, AB and O, right? This is the 4 types of blood group we know. And we divide the blood group depending on the presence of antigen, right? So I write here present of antigen, right? So if you have the blood group A, so how we represent? We represent like that. So we all know in a blood cell, on the outer region of the blood cell is also present the antigen. If A blood group, it have A antigen, right? If B blood group, it have the B antigen present on the outer side, right? So in the same way, if we have the blood group AB, that means we have 2 antigen, right? So I write here in the same way. Suppose this is A and this is B antigen, okay? But in case of O, we all know that O is exceptional, right? In case of O, they have no antigen, right? So I write here as only this cell. Here on the outer surface, no antigen also be present there. This is exceptional, right? All we know. So today we talk about the Bombay blood group. It is same like O, but it's not O. Actually, what happened, I just tell you that in the Bombay, one person also be discovered that they have the blood group, it's have the antigen, but this antigen is not functional. So if we understand this phenomena, we have to understand very clearly what is actually happening, how we decide that which blood group we have, which blood group express we have. So let's do this. So suppose all we know that the chromosome, right? Chromosome, and within the chromosome we all know that so many genes are there, right? So many genes are responsible for the so many character. So this blood group also be character, right? Suppose if we have the A blood group, that means there is some, we all know there is some gene also be present for this character of blood group A, right? So this gene also be responsible for the A blood group. That's I write here the A gene. I write here IA. I means immunoglobulin A. Okay? This is one type of gene which is responsible for the A blood group. If gene is expressed, then your blood group also be A. Okay? So in the same way, if your blood group may be B, okay? So here, this is also be gene also be present in your body of a cell. That is I write here IB gene. If gene also be expressed, then your blood group also be B. So you all know that in our all character of our body, all we get from our parents, right? Suppose parents means father and mother. Some character also be transformed to us from the father, and some character transformed to us from the mother, right? But all characters are not to be expressed. Some character expressed and some are not, right? So that's why we all different to each other. So here, if our blood group also be A, there is also be gene also present, that is called IA gene, which is responsible for the A blood group. Okay, A blood group means this gene is responsible for the antigen A. I write here antigen A, right? If this gene also be expressed in your body, then this gene produce the protein like antigen A. And if you have antigen A, that means you have the blood group A. In the same way, if your gene also be expressed in B, then you have to produce the B antigen, right? Clear? Understood? So in this way, suppose if I tell you that if you have the AB blood group, what you say? You say, sir, simply, it is very easy. AB blood means we have both the genes also be present, right? Both the genes present means we have both the antigen also we like to produce. That's why our blood group also be AB. That is the main phenomena. Okay? But there is a twist. Twist means these genes also be expressed when there is another genes also be present. It's when it's also be active those genes, then it's also be expressed. We all know the genetics, we all know the concept epistasis. You know very well epistasis. Epistasis means this is a phenomena where one gene also be blocked to the other gene expression. Suppose if I draw here, suppose this is a one type of chromosome. Suppose within the chromosome, this is a one type of gene, this is another type of gene. If the gene is also be expressed when this gene also be active, so that means the B gene activation is depends on A, right? So in the same way, our antigen gene also be controlled by other genes, and this gene also be called, I write here on the bottom, that is called another antigen, that is called H antigen. Okay, H antigen. This is one type of antigen. Okay, I write here FUT1. This is the name of the H antigen. Okay, and this antigen also be produced by gene that is called H gene. Okay, so how I say that clearly these genes is directly responsible for the expression of A antigen and B antigen. That's mean if you have A antigen also be present in your body, that means you can sure that the H gene, or we like to say H antigen, also be present in your body. Okay, that means the H antigen directly, or you like to say indirectly, totally control the A gene and B gene. Okay, that means if you have AB blood group, that means you have both the genes also be present, A and B, and both the genes also be controlled by H gene. Now you will just say, if your H gene also be defective, just I write here, if it is defect, somehow it is defect, then what happen? What happens, sir? If this gene also be defect, then what happen? This gene be expressed or not? No, sir. If the gene, this H gene also be not expressed or non-active, then the A and B gene are not expressed, right? So in that phenomena happen in case of Bombay phenotype. So what we see, the scientists shocked that the patient which are also be treated as Bombay phenotype or Bombay blood group, what happened? They have A gene and B gene also be present, but those genes are inactive. Why? Because their H gene, or you like to say H antigen, is defective. If the H gene is defective, then what happened? The A gene or B gene, if present or not, it are, what I say, I just write here, A and B G antigen are non-functional. Okay. If you have A gene or B gene also be present in your body, it is non-functional when the H gene also be inactive. That means H gene is so many importance, very very important for the activation of A gene and B gene. Clear? So that is the Bombay phenotype. We all know that in our blood group O, they have no antigen, right? So that's why they are the universal donor, right? So in the same character observed in case of Bombay phenotype, because it have obviously it have the A gene and B gene, but it is not functional. That's why the Bombay phenotype also be treated as a O blood group. Okay. So now you have the questions arise in your mind that, so how, if any patient have the O blood, Bombay blood group, then from where they can take in the blood when they needs blood, who gives first? It is same like O. We all know the O is the universal donor, right? They can donate anyone, any blood group, and he have that when he needs blood, he have the blood to take from the only O. So in same way, the Bombay blood group patients takes blood only from the O, same like O, but it's not the O. Okay, that is the main phenomena of the Bombay blood group. It is actually discovered in 1952. Okay, I think you just clear. This is the main short concepts. If you like this video, please subscribe my channel. Please stay with us. Thank you.